All right, well, welcome everybody. Thank you all for being here for today's webinar hosted by the Arkansas Native Plant Society. This webinar is being recorded. Afterwards, the recording will be uploaded to our YouTube channel. The mission of the Arkansas Native Plant Society is to promote the preservation, conservation, and enjoyment of the native plants in our natural state, and to educate the public regarding the value of these native plants and their habitats. If you'd like to learn more about the Arkansas Native Plant Society, you can do so by visiting our website at anps.org. You can check us out on Facebook at facebook.com/Arkansas Native Plant Society, or you can always reach out to me by email at anps.president@gmail.com. You can get on our email list to learn about upcoming webinars that we have planned in the coming months. So I'd also like to encourage you to consider joining the Arkansas Native Plant Society, and you can do that on our website through PayPal. Just go to anps.org/join. To give a quick plug for the next two webinars coming up in our series, Wednesday, September 4th at 1 p.m., Dr. Richard Abbott is going to talk to us about the native vines of Arkansas. And on Wednesday, October 6th at 11 a.m., Dr. Travis Marsico will talk about the important role of herbaria in recent discoveries of invasive species risk from hitchhiking propagules at international seaports. Our speaker today is Dr. Subedi. Dr. Subedi is an assistant professor of biology and herbarium curator at Arkansas Tech University in Russellville. His research focuses on biodiversity patterns, community structures, ecosystem functions, disturbance ecology, and ecological monitoring. He did his PhD at Florida International University, did his dissertation on functional trait approach to examine plant community dynamics in South Florida hardwood hammock forests. Today, Dr. Subedi is going to present to us on oak diversity in Arkansas. So with that, I'll hand it over to Dr. Subedi.
All right, thank Eric. So thank you everyone to be here. So I'm excited to talk about some of the stuffs that I'm doing. So I'm talking about the oak diversity in Arkansas today. So just gonna give you the outline because there are a lot of stuffs because of considering the audience that I expected to have knowledge, different levels of knowledge in botany or plants. So first, I'm trying to give you the introduction to the oak family as a plant family, the Fagaceae, and then I'll give you some general characteristic of the oaks, how we identify them, or how we say these are oak species, and then I'm gonna give you some idea about the taxonomic classification of the oak species. So how many species out there, and also talk about the diversity in Arkansas. So, and then with that, and then I'll give you some of my ideas that what I'm trying to actually answer the research questions, my student involved in it, and then my interest or what I'm going. So, but I just want to make sure that when I'm talking these results, these are preliminary results. So I'm still working on it and they are not peer reviewed. So I'll just share some of the results that I have got right now and then the future direction that I'm heading to. So with that, let me introduce to the family. The oak family is in Fagaceae, like I said, and then it's very interesting to see the oaks are only in the northern hemisphere. So you don't see them in the southern hemispheres or southern hemisphere. So you see the all red, that's in the north. So generally, most of these species are in North America and then Southeast Asia, then few are in Europe, but all of them you can see there are a couple of species in the northern South America, and then there are a few kind of in the northern Africa, but most of them are in the northern hemisphere. So, and then there are not many genera, just 9 genera, but there are a lot of species, 900 species, and then actually it is very dominant family in terms of mostly the oaks are 80% of our vegetation are actually in this family. So generally, this is all trees and small trees, or few of them are shrubs, and a lot of tannins in it. They're really tough. Most of them, the leaves are, and trichomes, the hairy stalks on the leaf surface, or maybe some twigs are the important ID for the identification, and the leaves are always alternate, simple, and then there are many of them are low, but some of them are not. Margins are very entire, means very smooth to the serrate or dented, those kind of stuff. The venation, I don't think you need to know this more, but if you look at this under the microscope on the surface of these leaves and the twigs, you see different structures. So the taxonomists, people use these different structures, and which are very important characteristics. The trichomes are to identify different species and genus in this family. So just give you the brief, the flowers types, and then we call them catkin. It's kind of have all these flowers kind of group into one inflorescence, you see that. And these are monoecious, so they're both male and female flowers. They are separate, but they are in the same tree or individual. So sometimes they are separate out in inflorescence. Sometimes they are both together in the same inflorescence. But you can see, I'm going to talk a little about the cap, the cupule on the female flower. You can easily see it under here, very thick, scaly cupule on it. So the cupule or cap, generally you see when you see the acorns that's the one of the way that you identify the Quercus, the warp genus. And then you see also have other species, but generally sometimes you see these are spiny. Sometimes they are smooth or scaly, but the scaly ones are the one that in Quercus, the other two species, the other species that in that family do have the spiny ones. I'll come to that later. So in US, basically you see two species in Fabaceae. They are mostly in the western part of the country, and then the other two include they're exclusively in the eastern, the Fagus and Castanea. And then the Quercus is widely distributed across and 5 or 6 are in Canada, but mostly in United States and Mexico and Central America. So the Fabaceae family as a family, like I said, there are 4 or 5 species, 5 genera. And then in Arkansas, we have 3 genera. So we have the Castanea, Fagus, and Quercus. So, and then these 2 species in Castanea, and you have the 1 species, Fagus, and then Works have 27 native species. So that's why the spiny cupule outside of the acorn, that's how you identify all the 3 species, but the Quercus has just the cap. So this one, so the value of this work is big, not only in United States or in Arkansas, but all across the globe. So in Northern Hemisphere, they are mainly many societies, they use it as a sacred trees, symbols, or the different ways, culturally, historically, different values. And also they've been really good at, because they live very long, and then they grow also very slow. So they are very good at carbon sequestration, and then also reservoir of diversity. So they've been used for many other organisms as well, and they retain the soil and water. So they protect the landscape as well. So very important. And then also for economic point of view, they are used for furniture, cabinet making, all kinds of stuff. So it's very important genus. So how to identify the works? They all are shrubs or trees, and many are trees, and there's few small trees or shrubs. The leaves are very variable. You can see it. They can be in different shapes, different type of lobes, so that it's very confusing. Sometimes I'll come to that. They easily hybridize, and sometimes they mix, and then we don't know whether that's one species than another, But the acorns and the cap is very important for the identification. So you see the space that it's kind of covered the acorn kind of give you a lot of idea what its species actually did. So you see I'll come to that later. So another characteristics, the leaf is the bristle tips. So you see the extension of little bit of outside this, the veins outside of the leaf. It's called the bristle. So some of them have this thing on the leaf, some of them don't. So that's very important characteristic when we divide up the work. I'll talk to that later. But usually you see the mid vein at the middle, and then you have the margin. This is the margin, and we say sinuses. This is when they cut, and then you have the petiole, and then the midrib on the side. That's where mostly we see those trichomes, the hairy structures, and they're also patterns, and that's give you a lot of information for the identification as well. So when you look at the acorn, so it's pretty much very regular other dicot seed, but the cupule, the cap, or we say the scales, that's at the bottom and cover. Sometimes it's go all the way to the cover the whole seed, sometimes up to the half, sometimes 1/3, sometimes 2/3. So it depends. That's another characteristic very important for the identification. So the classification of this genus is still taxonomists or botanists or scientists, they are debating a lot of new information coming in of the molecular data and other stuff. But in general, we see this is a genus and there are two subgenus. That's why the Quercus and Suresh. And then you have these sections in its subgenus. So these are different. So mainly you have 8 here, 3 in Suresh, and then 5 in Quercus. And then this is kind of a phylogeny of this. So you see that different, this leaf patterns and mainly the fruit characteristics actually. It's pretty good that a lot of molecular data now actually kind of not changing a lot in the way that taxonomists actually classified this genus earlier. So kind of supporting a lot of this, but there's few changes, but not much. So the main thing that I want to show you is this. So how much we have the species of this work in the world. So globally it varies, so that sometimes the botanists or scientists, taxonomists, they give the new name, and sometimes the other gonna argue with it, but they're around, we think, 400 to 500 species. So in North America, it's a huge number, it's like 245, and then Eurasia about 200 species, but not many in Europe. So there are a couple of things. The lobati, this is a red wax. I'll come to this subgenus. This is a section actually, kind of only present in America. So there's no this in Europe and Asia. So, and then most of this high diversity of this genus is actually in between 15 degree to 30 degree northern latitude. So most of the species in there. So if I put that in the table, kind of in a way in numbers, then and look at the Arkansas, we have 2 groups only. The white wax, 10 species, and then the red wax group of 17 species. So total of 27 native species. So actually that's very high diversity in terms of if you look at only in the state. And then, so I think this could be the very interesting thing that I'm looking at is, so when actually this species, actually evolved, and then how it diversified in these, only in the northern hemispheres of these 3 continents, the Europe, Asia, and North America. So there are 2, I don't want to go in detail all of these things. There are fossil remains found in all 3 continents. And then the oldest one, as far as I know, there are a few I saw in the new papers. I think it's still debatable, but this is the one that mostly people talk 55 million years ago. So the earliest one in Europe they found. And then the main thing is how they diversified in these 3 continents. So there are 2 theories appear to be. The one, these theories are, probably is evolved in Southeast Asia, right here, and then goes and went in 2 directions. So one toward the Europe, the another toward the North America, and then there were land bridges at that time, and then eventually they diversified in different places. So another theory is actually it used to be a same continent. So, and then there's a continuous population of that species all across these 3 continents, and then this continent are separated out because of the continental shift and other reasons, and then now turn out to be a separate population, and they diversified over the time in different places. So there are 2 actually theories. So I believe in the second one more, because it looks like a lot of diversity all across, and there's exclusive groups both places in East Asia and also North America. So they could be possible that it's kind of have a lot of fossil. There are a lot of many fossil records out there. So it's possible that it used to be a continuous one population and eventually is diversified. So now I want to come back to the 2 groups that we have in Arkansas, the red oaks and then the white oaks. So these are the really some characteristics that you can easily look at it and identify which groups that particular Quercus belongs to. Generally, the bark are darker and fissured in red ones, and then the white groups, they have a lighter slightly and scaly, and then the leaves with the bristles. Like, remember I showed you earlier the extension of the veins outside the leaf, so at the tips. So if you see that, that's belong to the red oaks group, and then if you don't see that, that's in the white groups. And then this is another interesting one, is in red oaks, actually the seed germinate in 2 years. It's mature in 2 years. Where the white oaks actually in 6 months is ready, and then they started to germinate. So within 1 year, you see that the nuts is ready to germinate. So the calves is a thin scales, where this is more thick, and then a little bit of softer. This is a little bit of harder. And the seeds, this is another characteristic that a lot of actually animals, they prepare to the white oaks, because the red oaks are bitter. They're not, so they are little not so bitter, because of a lot of tannins in the red oaks, where there's not much in the white oaks. So if there is options, that's what some of the literature are saying, that you have the squirrel around, and if you have the white and red oaks together in the same community or same place, forest, there's a lot of squirrels, they crowded, or the birds or other things crowded around the white oaks, and they eating the acorns. So, but there's another theory also that some birds or some squirrels actually they kind of they kind of herding this for the spring. So and then eventually over the time, the tannins in the red oaks going to go away, and then it's they think that they can eat it. So that's what the difference is. You see the bristles coming out of the leaves, and then when you see the Quercus, the white oaks, it's kind of more smooth, and you don't see that easily. You can differentiate the 2 groups. So another interesting stuff about this oak is the hybridization. So they can easily hybridize to each other, but they are exclusively within their section. So white gonna hybridize to the white group oaks, where the red gonna hybridize the red group oaks. So they in situ or out in the natural environment, they don't hybridize across these groups. So that's why sometimes you see that the works could be look like one species then another within the same species, same group, but not across the other groups. So this is the one that you see that probably the white work and then the post work. So they are in the same group, so they can easily hybridize and give you sometimes confusions which one is that group. So now this is book recently published, a very good book, and then it has reported 27 native species in Arkansas, and then we have a 10 white oaks and then 17 red oak species. These red oaks are which are exclusively to Americas. You don't find them in Europe and Asia. So and then there are a few species are concerned in terms of their population. So their population are very low. So these are the ones that they already kind of have, you know, we are all concerned about their population, and we try our best to preserve this species, or even we can try to increase the population as much as possible. So I'll give you the list of this 17 native Arkansas species. You have, if you are familiar with the common names, you know, the maple leaf is a actually this could be the endangered species. Arkansas oak, another concern, conservative species. There are a lot of diversity in this group in Arkansas, where we have 10 species from the white oaks group, and then these are the ones that you see around. So like I said, it's easy to identify the groups. You see the bristles around, and if you see the acorns, we can easily say these are red oaks, and then the white oaks, you don't see those, and then you can easily see from the bark, acorn, and the leaves, they are the white group species. Same with the red group. So now I just want to share some of my work and then the things that I'm interested in, and those are all background. So I'm curious because it's just like this first summer I'm here in Arkansas, and then I see a lot of this oak species around, and then I'm really interested to work something. And there are a few concerned species in terms of their conservation. And then I want to know, okay, where are the high diverse areas in Arkansas? And then what are the factors associated with this high diverse areas? And then I just want to know also, oh, there are so many species, like 27 native species out there, and there are a few conservation concern species. So I just want to know what is the functional and ecological needs of those species, and how their population went down, and why, if there is any way that we can understand what is the requirement for those species. At the same time, a lot of things are changing over the years, and it's going to change in the future. For instance, the climate change is one of the issues that we are talking about. These climatic patterns are kind of really random now, and a lot of temperature going up now, and then could be longer dry season, could be longer wet season, we don't know. So how they cope with this future changes. So what would be the scenarios if things go either way or other way, something like that. So I, from my work, research work, and then my, in a way, my little bit of expertise. So I just want to know, finding out regional and local factors, and mainly the natural factors like climatic, edaphic, like a soil factors, topographic factors, how that actually kind of driving this different species distribution patterns of the oak species. And then another way that I want to look at it is their ability to tolerance, or their ability to adapt to the different condition, is just measuring the functional trait of those species in different scales. Like in the populations, there's a different way that, there's some plasticity kind of a lot of variation, wide variation within the same population, and there could be, there's a lot of differences of between the species, and at the same time, the same different species in different community may be responding differently. And there are some dominant species, and there are some rare species within the community, and how that community actually cope with the different environment, something like that. So, and another interesting stuff that I just introduced a little bit of it is biogeography. Okay, so where are these species, and historically where they were been, and then where are now, and how the things are changed over the time, and what's going to happen likely to be in the future, something like that. Then we can look at it, the evolutionary history of those species, and also the biogeography, then give the idea that how we can understand their distribution very well, so that we can use the main objective of all these different approaches is all those things is mainly the conservation and management of those species, and as a whole the group. So that's what the main my questions, most of my research, and then I'm trying to do the same with the work species as well. So I just want to introduce some of the basic stuffs, because without knowing this, it's very hard to understand what are the regional factors, what are the local factors, and whether that's a population level, or that's a community level, or that's kind of more at individual level. So this is a very good example of it. What are the processes across a different ecological scale? So if you're looking at, if you want to do, what's going on at the regional scale, so you're mostly looking at the climatic factors. So that's the one that driving the differences at regional scales. If you want to do it at the community level, so that could be both regional as well as the community factor. So community factor may be soil differences, the moisture differences, the temperature differences, those kind of stuffs. And then if you look at the population level, like a species, between the species, then you can look at it, compare, what state is of this forest going to be, sometimes that dry, and then also community, like the factors I already mentioned, those also very important. Some species can through the dry condition, some species may not, or some species can be very good at the acidic soil or calcareous soil, so those kind of stuff. And some species may be good at high elevation, some species may not be good at that elevation, something like that, or topography. So the individuals is more like, you are talking about the more like a genetical composition of the species. So how that it be good, how that evolve over the years. So for instance, you can give easily examples of a water lily in water. If you put that in dry environment, you're not good. So those kind of stuff. So, and then there are other things also. You can look at the leaf and stem levels, cell level studies. So there could be different way that you can look at it. So for this thing, I already tested in other places. So I think this is going to be a very useful for work species also. Looking at the diversity, and then just looking at the regional scale, the factors I'm talking about. So that's kind of filter out maybe those 27 species, whatever we have, the work species in this region. So this is just I'm explaining today for this Arkansas state, but my study going to be likely going to be big. So so that's what, please don't confuse with that. So, but this is going to be easily going to be applied to any places. So, and then you come up to the habitat scale, the community scale, then that particular habitat going to filter out those species based on their trait. And when you actually within the community in a small scale, those are like a local species pool, then they try to compete to each other for the resources. So that's how the basic stuff. That's why all this trait, the functional trait, whatever they have, going to give us the idea actually how they actually assemble those species in that location or community. So just going into the basic stuff now. So I just look at in general, this is not from the data. So where are these different species, 27 that the native species in the work, and then based on in general, So I think there are not many in a really dry rocky upland. There's only one. I think that's the one that the maple leaf wolf with the endanger. And then you have another one is mainly concerning species are in sand barrens, very dry environment. Sometimes they are saline environment in the southern side. So and then the others are very common, like a white oak or red oaks. They're all gonna, distribute. You can find them in the upland areas, in the flatwood or bottomland, riparian forest or roadside. So those are the places they are very common. But you see the numbers; these are the species number. But I see this two habitat, if they are specialists on those places and they have a hard time, probably that is the reasons that that something has to do with this. And then what I'm doing right now is, because I'm really interested in not only looking at the present distribution of the species, but I just want to look at also how things change, where actually those species were reported from. So my best thought gonna be, I just want to look at the herbarium collections. So I just want to, and there are a lot of herbarium collections over the hundreds or 200 years, and they've been deposited in different parts of the country. And then I can extract, I can gather all those whatever herbarium. In this case, this project, I and my student doing is we assemble all the herbarium collections from Arkansas. So we start that, and then we're going to do is a geo-reference that data. So some of them have already have the location, latitude and longitude. Some of them, they don't have it. Many of them actually, there are 10% or 15% might have, but not many of them. And it's very complicated stuff also to get to the lot of information that they're probably not going to give you the accurate data. There are a lot of challenges also. So, but there is a process to do those. And then once we have those georeference data, then we can extract all those things that I was talking earlier easily, the topography. So what elevation, what slope, what aspect of that species were collected from. And then we can also get the climatic data easily available now. So of that particular location is more regional. The climatic data is really precise, and the soil data probably we can get also of that locations where the species were actually collected. So now that concept, what we are doing now is we have the SORNIC, the Southeast Regional Network Expertise and Collections, and our university is also part of it. So there are like 233 or more herbaria actually involved in that institutions, and there are 200 years or more than 200 years of collections out there. And then we have also a lot of collection of the work species in our university. Some of them are already included in that SORNIC database. So, and then first we looked at it all in this SORNIC network, that how many work species actually reported, actually collected from the Arkansas. That's what the first step we started with. And then we filter out all the Quercus species in this state. And then we try to georeference them, the latitude and longitude. I think most of this work usually use the geolocate software. So it's very similar to the Google map or other things, so you just have to put the comments, whatever. Oh, okay, this road 30 miles off toward the north, and then that's where the species, the specimen was collected. And then we try to get as much as close as possible based on the comments in the herbarium, and then we get that lat long. So, and then that's how we use to extract those other data, the factors that we are talking about, the climatic factors, the soil factors, and then the landscape, the other like elevation, slope, aspect, those kind of stuff. So far, what we found is there are about 6,000 records of work species altogether, all native species. So we exclude all the non-native and exotic species and hybrid also. So you can see that out of 6,000, 4,820 records were native species. That's how they distribute. You see that here, there are how many records for each species of them. So for instance, a lot of them are, they still at a postal. And then the southern redwood, the falcata, you see that all tall. And then there are hundreds and more for the maple leaf oak, and then there are about 150 or something for the Arkansas, another important species for conservation point of view. So this is very good that, and then we exclude all this, and there are a few never identified to the species level. There were like 300 records or so. They just say the Quercus species, and then there are non-native of 750 records that we found. And then there are a lot of hybrids also out there. So, I mean, not many, actually, there are 120, about 120 records. And we looked at it, actually, my student and I looked at it. We're still in this process though. And what are the combination of that? You know, the textbook we're saying, well, there are not, I mean, there's still a, you know, the identification of the species is questionable because there are some are expert already determine those species, but some of them are not. So, but we try to look at it, the combination, and we usually see there are red, white. So there are not many 120, but still that's there. So the combination of the hybrid, whatever they assign to the herbarium are not mixed with the red and white. So far, this is kind of snapshot of the locations we found. We kind of have idea now. We're still working on it, the error part of it, but out of those, so far, with my students, we determined that there are 2,593 records. We geolocated them, and in Arkansas, these are the locations for all the species, 27 species, native species. And then we also have where actually for each species located. For instance, this one is a prinoids. You see that they are in different counties. So these are the ever recorded, like historical, wherever they collected the specimens from. So the Quercus Arkansas, one of the species, you have this, the herbariums actually collected from this many places. So another interesting stuff about this, this process is, like, this is just from that book, and this is kind of a conservation concern species that from Arkansas is highlighted 2 counties where actually is reported that species. I believe this is a current kind of present of that species, but I looked at it, the historical data, and then I can see that in 1973, and then 1970, 1957, 1932, they were reported from different, you know, it looks like it has a wide population used to be. So that's kind of, this is the work that I want to see how, you know, this the population of each species change over the time. Also, we can look at it. I mean, there could be a bias that, okay, some place will go and collect it, but it's going to give us the idea. If someone is collected from this place and this corner and that place, we can assume that it will have a wider distribution across. So based on that geolocation data, so we extract the topographic variables. That's what we've been doing. So we have a really good LiDAR data that we can get from USGS and other sources, very high resolution data. And this is the one that we calculate the slope, we calculate the aspect. You can see that northern aspect or southern aspect, flat, east, west, southeast. So we have a, and then we also calculate the slope, actually where that species were collected from, and we have the elevation also. So if you put in the elevation map, you can see that it's pretty random. So you can see that the collections were from one part, not another place. It's pretty random and all across the state. And then, so we haven't finished, but I'm going to present you today in a kind of one at a time, the elevation slope, but eventually we're going to put together all these topographic, climatic, and then soil variables and see where is the ecological needs of each species or high diversity areas. But this is kind of a example of the way that we've been doing. So you can see where the high diversity in terms of elevation. So the higher 25 species, 50 to 100 meter, and you see the similar pattern in the red and white group of species. And then if we see the 50, between the 50 to 300, this is the color area. So that's where the most of the species and many species actually occur in high diversity areas. So this is not only going to determine that we have to fit the model, that's we're going to do it later, but this is the idea of it. So it's similar with the slope. We can see it where actually what slope actually the high number of species actually reported from. So it's very similar concept. And then mostly if you want to see this 2 to 8 degree slope, actually most of these work species are from, and you see the number of species actually all almost 25 or 27 species from that areas. It's similar with that. The aspect is more flatter. So they are from, there's no only one aspect that you have a high diverse or many common species are from. So, but when we fit the model, then we'll see that what actually explains a lot of diversity in this. So that's what the one that I just want to, because of the time, I don't want to, I can't do it all, but I just showed you we do the same with the climatic variables, we do the same with the soil variables, and eventually we combine all in the model, and we'll see the species distribution modeling, and then we'll show that where actually the high diversity, what kind of ecological needs actually the best for the high diverse work species, and then we can also test with the test to the each species that if you were interested in. So the second aspect that I was talking earlier is a functional diversity and the species distribution. So I don't want to go in detail. The trait or any description, any morphological, physiological, or life history characteristics related to the fitness of the species. It's been recently been used a lot in different areas and very good, like a specific leaf area, the thickness of the leaf, there are different things, the heights of the leaf. That's going to explain a lot how the plant's going to do in particular location. So you can see the work species of different, they have a different seed type, they have a different shape type, the different sizes, and that has to have some meaning. And so if the plants or individual species is a plastic, sometimes they vary with the shape and size depending on the environment, then that might have a more ability to tolerate various conditions. So and there could be many traits we can select, and then we don't have to even measure all this now, because there are already in published papers that one of my colleagues actually showed that the herbarium-based measurement actually reliably, you can estimate the different functional traits. So we can do that with using the herbarium specimens and then try to estimate the plant's trait. So that's another way that I'm thinking about. So I'm going to show you a little bit of, if I have a time, about the trait, the seed that we are collecting. So now, the acorns are very popular among the wildlife. So you can have all kinds of wildlife here, the waterfowls, woodpeckers, the birds, the squirrels, they love it. And there are some terrestrial animals and so on. So, and they prefer one from another. There are some literature that I read and then found out very interesting. So, and then I looked at it myself in different community, and you see that some community do have a lot of sapling and seedling under this work trees, and some they don't have it. Even I don't see even different age. Like I tried to see even 10 years or 12 years, 5 years, and they are not there. There are big trees around and then nothing underneath. So what's going on? That's very interesting. And so, and there are some, like I said earlier, and some literature are saying, okay, white works, the birds or animals they prefer because it's sweeter or not bitter. then there are some literature saying that actually they go with the familiarity is a general component. So if they have the red works around for a long time, they used to go with the acorns with the red works rather than they go to different ones. So that could be possible that if something is introduced to the area, so the non-native species may have advantage. That's another thing that we can look at it. So and then we put together all this. This is the one that we already finished, all the acorn collected, and we have already, some of them are already here in our herbarium. So all these species, and then I'm interested to see the size, because they're saying the site dependent dispersal, you know, so some I think there are both schools of thought there. So some said, oh, small sizes acorns disperse long distances because the bird can carry those and prefer the small sizes. With the animals, they usually prefer the bigger ones and they not go far from the tree. Whereas some of say, oh, the bird actually they prefer the bigger one, the acorn. So, but still we all have this characteristic, and it's very cool that if you think about the seed size is an important factor. So we're going to do the size. These are based on the seed weight though. My student collected a few and we already have them, and they are from at least 10 different populations, and we try to get whether that's have any pattern on it with the geographical distribution. So this is still in progress. So, but we'll tie this with all the other the leaf characteristics, the wood characteristics, like the wood density is going to give you the drought resistance of the tolerant of the species, and that, or the plasticity between within that particular trait, and then compare with within a species and between species. You know, that's another way that we can look at it. So now I'm a little bit out of time, so I just want to say you the what actually I'm trying to complete soon is to complete the geolocating work. So just try to finish this 5,000, 6,000 species that we have the list, and then also add more data from other herbaria as well as from other like iNaturalist or other resources that we can get, and then do the same the topography as well as the climatic and soil data, and then fit the model, the species distribution model, and see what actually have a big role to play in terms of explaining those diversity. And then also try to gather the functional trait as much as possible using both herbarium resources as well as the sampling the trees. You know, there are not many trees, just 27 species. And then let's see if we can come up with some idea and then predict their distribution in future with the various scenarios like temperature changes or rainfall changes or various kind of scenarios. Oh, this much going to change what happened, or this much going to change what happened, something like that. So I want to conclude with this, and some of the students involved in this project, most of the work were done by my student, and they still doing it. And then there's little funding coming from the Arkansas Division of Higher Education for my students, and then some funding from my university, Arkansas Tech University. So I would like to thank to all of them. So I'm gonna stop here, and then I'll happy to take if you have any questions.
All right, thank you, Dr. Subedi. Let's see, I know we had a few questions here in the chat. So, and Brian, if you wouldn't mind re-entering your question, my computer died and I had to get on my phone, and so I'm not able to go back and see the questions that were in there. I know there was one question about the species of conservation concern. There was a listener who wasn't able to write those down fast enough, and could you just repeat those, please? Are you able to hear me?
Oh, you're asking me?
Yes, yes. Where you had mentioned some species of conservation concern earlier in the presentation.
Oh, I see. Yeah,
sorry.
Yeah. I mean, that's a one thing that I refer to the book.
That's
This
okay.
one. Yeah. So this is the one that I looked at it, but that's actually, I got those information from the book, this book, you know. So if you look at that book, have a lot of details, and then counties actually, you know, said it. So where actually that's only limited to only like a couple of counties or 3 or 4, those areas, you know. So these are the concerned species they reported. I know that Acerifolia and then Arkansas have also the concern. I mean, it's in the red. The IUCN put that in the red list also. So, and then this one, the Darlington Walk and Dwarf Chestnut Walk, and then Durant White Walk. These are the one that conservation concerned species that reported this book, you know. So that's one that if you buy this book, it's a pretty cool book, a lot of information about the oak species as well as the other plants, the trees, shrubs, and woody vines in Arkansas. Yeah. So there are some questions. So you reading it? So would it be possible? I can read it.
Sorry, I was muted. Yes, if you would like, go ahead.
Yeah, there's a question. Would it be possible to get the geo database with the geolocated work trees? Yes, eventually we're gonna provide it to everyone. You know, I'll share with the Sarnic actually, you know, so because that's a good thing to, that's a lot of students doing. And then once we have completed it, and then we try to maybe publish things that we get out of something, publication from the project, and then it will be available to the public. Yeah, it will be available. Let's see, there are a lot of questions. Have you seen more white wolves dying in Arkansas during the past few years? All right, so that's one of the things that, you know, I don't know, you know, because that's what I'm not doing research, what is species dying right now. But the main thing that I'm trying to understand in a bigger picture is, okay, what are the ecological needs? What are the requirements for each species? What is the best? And then how much things change over the time, and what actually driving the high diversity, and as well as the species, each species. So I don't know right now. I mean, if white wolves are dying, that could be the one that predators play the role, or habitat, you know. Actually depends on what species you are talking about. So I don't know exact answer for that one. So another one, do you consider cherry wolf a separate species, or is a variety? It's a separate species, you know. It's like that book have already, and then Quercus pagoda is a separate species, and then southern red wolf, the Quercus falcata, is a separate species, I believe.
I think Brian will be happy to know that, since he says his license plate is Q Pagoda, so don't have to change that yet, Brian.
Oh, my truck license plate.
And to briefly answer Martha's question about a copy of the PowerPoint, I mean, this will be up on our YouTube channel to watch later, but I don't know if you're willing to make a copy of your PowerPoint available, then that's up between you and Martha, I guess.
Oh yeah, I think I'll probably make a PDF and send it to you over in the email so that you can supply to them. So
okay,
fine. So
yeah, well, email me then, or all the listeners who are wanting a copy of the PowerPoint, send me an email at amps.president@gmail.com, because I don't have your email addresses, since we just sent the link out to a bunch of different locations. So if you would like a copy of the PowerPoint, just reach out, and I'll forward that on from Dr. Subedi.
Well, there's a question here. For your information, many of those prinoids records are actually movers. Yeah. Okay. I mean, right now, I'm not doing the questioning. I know that there are a lot of questions. There's a lot of work to do, the identification, because I looked at it, the North American, all the work she put it, there are like more than 200,000, even more work species, you know. So there's people, the folks are working on it to correctly identify those species, you know. But now, because we have so much collections out there, so we just try to get to in general, you know, just not going with the one location or one specimens rather than in general. And then once we get to that point, or if something is questionable, like he mentioned something like prinoids, ecominata or something, then we'll go to those species later, you know. So for now, it's just geolocating those. I mean, even if that the different species, we need to geolocate them, you know, so that where actually that collected from, and get those information, the environmental information for that species.
Okay. Let's see. Got a lot of comments on, and people really enjoyed the webinar. Looks like Jim Sullivan asks, will your model be statistical or tabular like the Braun-Blanquet model?
No, it's going to be very statistical, you know, that's what I do. So it's just for the presentation, we don't have enough information now to run the model, you know, because still we're gathering a lot of information, like I mentioned, climatic variables, also the soil information, and you're still working on finishing those geolocating the specimens. Once we have all those information ready, and then we do the statistical modeling, you know, it's not the tabular form. Yeah.
And Rob King is asking what the source of the color leaf images was.
Which color leaf?
You might need to expand on that.
Oh, the one that with the color leaf. Yeah, this one. Yeah, this is from the picture by this guy. I don't know him, but I found it in he's probably gonna be referring this one, you think, Eric?
Probably, yeah.
So I wrote it here, and then I found it in Quercus. There's what they call yeah, it's kind of, I have a lot of information international. I forget the name, but I found it somewhere there, you know. So I put the name there. Someone make a pictures, and I put that. So.
Well, that looks like all of our questions. Yeah, I really appreciate this. This was a really great presentation. I really enjoyed it, and I know a lot of other people did as well. Looks like there was a lot of great comments there in the chat, thanking you for the presentation. So we definitely appreciate you sharing your knowledge, expertise, and giving us your time for this. Like I mentioned, this will be posted up on the YouTube channel, and I'll send you a link, Dr. Subedi, so that you can share that with your colleagues if you wish to.
All right.
Just to let everyone know, the next webinar coming up next month is going to be the native vines of Arkansas with Dr. Richard Abbott. And if you'd like to join the Native Plant Society, you can do so at amps.org/join. So again, thank you, everybody. I really appreciate you being here, and we'll see you next month.
All right, great. Thanks.
Yeah, thank you.